And the Griffith experiment, so he had these dead mice, and so they recovered the S strain from dead mice. So they also said it's DNA, not protein. But up until this point, they didn't really have a definitive answer. Like they couldn't say 100% that it was DNA. Like they couldn't hold it in their hand and say, this is DNA, this is what causes inheritance, and this is what holds our genes. So Hershey and Chase, they actually were the ones that finally said that DNA is the actual, DNA is the genetic factor. They used bacteriophages. Bacteriophages are viruses that infect bacteria. And so they had radioactive sulfur and phosphorus, which attached to protein, and it attached to nucleic acid. I forget which one was which. But anyway, they could track where the DNA went. So a little bit about a bacteriophage. Bacteriophage looks like this, right? So the outer coat is protein. Everything in red here is protein. And then inside is DNA. And so they could track where the DNA went through tracking the radioactivity. Because the DNA attracted sulfur, I don't know which one, we'll say it's sulfur. So DNA attracted sulfur, they could track where the sulfur went. Protein attracted phosphorus, and they could track where the phosphorus went. So Hershey and Chase were finally the two people that could say DNA is the genetic factor. And then Watson and Crick came along, and they discovered the structure of DNA with the help of Rosalind Franklin, who was also part of that. So Rosalind Franklin kind of got shafted away from a Nobel Prize. She died. You can't give a Nobel Prize to somebody who's dead. So Watson and Crick received the Nobel Prize for discovering the actual structure of DNA. Before they could figure out the structure of DNA, Chargaff figured out the A to T, C to G rule. So the percent of A equals the percent of T, and then the same for the others. Percent of C equals the percent of G. And what he ended up finding out, in addition to that rule, it's a very important rule, he found out that every individual of a species has the same percentages. So Bozeman showed you octopus, and it was like 32% to 32%, and I think it was like 18 and 18. My math might be off, but for every organism that belongs to the octopi species, the ones that he was looking at would have that amount of A and T and that amount of C and G. So humans, we all have the same percentages of A, T, C, and G, roughly the same. This person, or these two people here, Messelson and Stahl. So Messelson and Stahl came up with the idea that DNA replication is semi-conservative. DNA replication is semi-conservative. So when we make a new strand of DNA, there's one strand of old and one strand of new. So this might be the old, and this might be the new. And then we would have another set of the same thing. So we would have new and old. So the old strand gets ripped in half, and then we add one new strand to each of those to make two helices. So they figured out that it was semi-conservative. The other theories that, or hypotheses that they had, was that it was completely conservative, which is what this is showing you. So if it was completely conservative, this would be old and this would be new. That wasn't the case. And then another hypothesis that they had was a dispersive replication, which was like a banding pattern of old and new. So something like this is kind of strange. But so what they figured out was that that was the conclusion. There is a nice TED Talk that goes over, oops, no, wrong one. There is a nice video on YouTube that goes over Messelson and Stahl's experiment if you want to look at how they reached that conclusion, but we don't need to learn the experiment per se, just kind of what they found from their experiment. So semi-conservative replication. We're not going to get into replication today. We'll get into replication on Wednesday. But we are going to talk about DNA pairing and how DNA looks. So I hope we can do this part first before we get into how DNA looks. So using Chargaff's rule. So what is Chargaff's rule? A to T, C to G, and we're going to synthesize a strand of DNA. So maybe should have made these red and orange like our old and new on the last slide. But anyway, if we have a strand of DNA and we need to make a complementary strand, so the new strand is complementary. Complementary. This is old. And that is old. So how are we going to match them? If A matches with C, C matches with G if we use Chargaff's rule, what would we end up with? So if you can write on your screen, you can enter these in. Or how about this? I'll do, I'll put these in segments. So choose a segment to write on. How about that? There you go. So you can click your screen, you can use the handwriting feature, or you can use the text feature and match those complementary strands, or make a complementary strand to the original strand. So far, so good. Okay, so I entered in the new strand. Oh, nope, that's not right. I entered it on the wrong strand. Anyway, here's me being proactive. I did it wrong. All right, so yeah, so GGC looks great. Okay. CTA looks great. AAA, great. CTA, yep. Let's see, ATC, yep. GAT, good job. GAT, AG, yep. AT and G, yep. All right, awesome. So we made a new strand of DNA to go with our original strand in our semi-conservative fashion, right? And we have no errors, so that's great. So on the bottom strand, what you'll end up seeing is that the complementary from the original will look like the new strand, and the new strand will look like the old strand from up here. And then if we like continued these, they would be like zipped like that. So we would have like this thing called a replication bubble. So again, we're not going to get into the replication bubble today, but we did a good job at matching those strands of DNA. So there you go. So there's the answer on the next slide if you want to come back to it. All right. Let's talk about the structure of DNA. So DNA, we've already learned about nucleic acids, right? Back from chapter 3. Nucleic acids are polymers, which are made up of these nucleotide monomers. So nucleotides that are connected end to end make a nucleic acid, right? Nucleic acid is the polymer that we learned about in chapter 3. So it's one of those biomolecules, or those macromolecules from chapter 3. Nucleic acids can be either DNA or RNA, or they can be ATP, but we're just learning about DNA and RNA. So DNA and RNA, they are a type of nucleic acid, DNA or RNA. DNA is found in the nucleus, the nucleoid region, or in some organelles. So you might be thinking, well, how can it be found in all those places? All right, there we go. So DNA, if it is a linear linear chromosome, or if it's organized in like a linear fashion, it will be found in the nucleus of eukaryotic cells. Okay. If we're talking about circular DNA, it will be found in the nucleoid region of prokaryotic cells. So remember, prokaryotic cells, they do not have a nucleus, right? They don't have an organelle that encloses their DNA. It's just found in a region called the nucleoid. Now, some of our organelles have little bits of DNA in them, so that would be like the mitochondria. And then if we're talking about plant cells, that would be the chloroplast. They have some bits of DNA left over. Those bits of DNA help regulate processes that are exclusive to that organelle, so that's why they still contain some DNA. And remember, mitochondria and chloroplasts are likely bacteria that cells took up a really long time ago through something called symbiosis. So they retain some of that DNA. All right, so that's where we can find DNA. So RNA can be found throughout the cell. So it can be found in the nucleus, it can be found in organelles, it can be found in the cytoplasm. And that helps to relay messages from DNA to other parts of the cell. So we're not going to talk much about RNA this week. We'll talk about RNA mostly next week. But just to let you understand the difference between the two, and since we're going over nucleotides, I figured we would talk about it. So there are different types of RNA. We're not going to learn about them right now. DNA contains genes, which code for your proteins by the help of RNA. So we already learned about genes and Mendelian genetics last few weeks. So now we're kind of getting into what is DNA, what are genes, looking closely at them. All right, so nucleotides are made up of three parts. One of them is a phosphate. Another part is a 5-carbon sugar. And then another part is a nitrogenous base. So let's talk about the phosphate first. So each nucleotide contains at least one phosphate. It might have upwards of 2 or 3 phosphates attached to it, but if we're looking at a nucleotide on a DNA strand, it would have 1 phosphate. That's how they would be found. The phosphate is connected to this 5-carbon sugar. So in DNA, the 5-carbon sugar is a deoxyribose. So that's what the D stands for in DNA. And then it's called ribose in RNA. The difference between the two is an oxygen. So ribose contains an oxygen, an extra oxygen. Deoxyribose has lost that oxygen, hence the name. This sugar, you'll see these numbers on it. They're pronounced as like 5', 4', 3', 2', 1'. And they just help tell us the location of the carbons on that sugar, and they help us learn like the attachment sites for certain things. So if you look, the phosphate here is attached to the carbon, or the 5-carbon sugar, through the 5' carbon. And then the nitrogenous base is attached to the 1' carbon. So this nitrogenous base can be one of 4 things if we're talking about DNA. It can be one of 4 things. The one that you're looking at here is adenine. And the other 3, so this here, this is adenine. The other 3 can be thymine, cytosine, and guanine. And then in RNA, we have uracil instead of thymine. Okay, I'm going to switch to the next slide. Are there any questions yet? We're going to talk about how these are arranged into a polymer. So again, we are looking at a monomer, right? So this is a nucleotide here made up of 3 parts. So remember when we learned about like fatty acids, they had like this glycerol part, and then they had like these fatty acid chains. So they were made up of like 4 parts. This nucleotide just has 3 parts, right? Phosphate, carbon, sugar, and a nitrogen base. So that nitrogen base is what makes the nucleotide different. When we talk about our A, T, C, and G, A, T, C, and G still have these parts connected to them. We don't bother saying those parts though, because they're always the same, no matter which base is attached to those parts. So we just say A, T, C, G. We know the sugar and the phosphate's always there. So what you'll notice is that the structure of these nitrogen bases is different from each other. Guanine and adenine are what we call purines. Let's see if I have it written. I don't. Purines. And the other 2, thymine and cytosine, are pyrimidines. The way I remember which one's bigger is that adenine and guanine have a 9, N-I-N-E, at the end of their name. And so that 9 is actually helpful because there are 9 atoms that make up those molecules. But anyway, the 9 just helps me realize that it's bigger, and that's the larger one. So purines have 2 rings. They have a 5 carbon and they have a 5 atom and a 6 atom ring. And then the pyrimidines only have a 6 carbon ring. So some other things that you'll notice about it is that when A and T go together and C and G go together, there's these ... structures between the 2, right? Those ... structures are referring to, oh my goodness, if I could write, hydrogen bonds. See if I can type it. Those are hydrogen bonds. So hydrogen bonds are the weakest kind of a bond, or one of the weaker bonds that form, and they form between separate molecules. So that's how water is held together. So water molecules are held together by hydrogen bonds. And what helps is that it keeps the structure together, right? So it keeps this like 3-dimensional structure together. But hydrogen bonds, since they're so weak, they're easily broken, and then we can copy a gene or make more DNA or RNA or whatever, and then put them back together and they'll reform. So when these hydrogen bonds form here, they form a double helix pattern. And the way that the double helix is made is called a right-handed model. So it's going up to the right. I don't know if you can notice that there, but it's going up to the right on all of these. So it's never going to go up to the left. I don't know why it forms that way. I think it has something to do with the bonding patterns between the backbones of the ladder, or the outside the vertical parts of the ladder. But I'm not quite sure why it's a right-handed model versus a left-handed model. But anyway, it's only ever found as a right-handed model. And let's see, what else didn't I go over? Right-handed model, it's a 3D helix. You might hear parts of the helix called, referred to as a ladder. So the vertical sides are made up of phosphates and sugars, whereas the rungs of the ladder are made up of the bases and the hydrogen bonds. So the vertical sides of the ladder are made up of the non-important parts, right? Phosphate and the sugar. So not really important, doesn't code for anything, just kind of adds protection. And then the rungs of the ladder, the insides, are made up of the important parts, the nitrogen bases that actually code for your genes. Let's get to it. All right, so this is our DNA replication figure. We're going to be on this figure for quite a while here today. Let's see. So first, DNA replication takes place during what phase of interphase? Might take you a second, but it takes place during S phase of interphase, right? So G1, remember, is gap 1. There's a little that goes on. The cell gets bigger. It regains some biomolecules. During S phase is when DNA is going to replicate, right? So we're going to make another strand of DNA so that during mitosis, the daughter cells can have the same copies of DNA, right? So during S phase is when all of this occurs, okay? Each of these things here are enzymes, right? So enzymes typically end in ASE. ASE, look at all of them. So these are all enzymes. Enzymes, remember, speed up reactions, and they might build or break down components. So if we look at all of these, what are they doing? DNA polymerase is building, DNA polymerase building, helicase is breaking, and this one's not doing either of the two, but we'll talk about that one in a second. So enzymes, again, they speed up reactions, and each enzyme here has a different job. We're going to talk about each of those different jobs. Okay. Let's see. Let's talk about helicase first. Helicase. So it's this enzyme here. Helicase breaks hydrogen bonds. So remember, each of the strands here, so the strand on this side, they're connected by weak hydrogen bonds. Each base is connected by a hydrogen bond here. And that hydrogen bond is broken by helicase. And so helicase breaks the hydrogen bonds so that these bases are exposed, and so that another enzyme can add new bases to the strand. Now, let's see, let me type up here. Helicase breaks hydrogen bonds. Okay. Okay, so let's go on to topoisomerase. Topoisomerase is that circular enzyme there, and you might be able to guess what it does. Topoisomerase prevents supercoiling. So remember, DNA is organized into coils. So it coils around histones, and then those histones coil around themselves, and they make this big condensed structure of a chromosome by supercoiling. Now, topoisomerase undoes the supercoiling so that the DNA strand is accessible by other enzymes. Okay, so if we write up here at the top, topoisomerase prevents supercoiling. You could also say that it like unwinds the DNA or uncoils the DNA, if that's simpler for you to remember. But I'm pretty sure you're going to see that supercoiling term in the textbook and the quiz that's coming up. So it might be better to write that definition instead. Okay, so now that topoisomerase has undone this supercoil and helicase has split the hydrogen bonds, what we're going to do next is, let's see, I'm running out of colors. Let's talk about primase. So primase, you can see is this like light blue ring here, and it's creating something, right? It's creating an RNA primer. So take a look at the colors here. This RNA primer is orange, whereas there's a red strand, right? So the original DNA backbone is going to be red. This RNA backbone is represented by this orange color because it's different. So primase makes an RNA primer, and that RNA primer is the attachment point for DNA polymerase 3. Okay, so primase creates RNA primers. So the RNA primer again is for DNA polymerase 3 to attach to. You can see there's a lot of primers here on this side, right? There's not that many primers on this side. It's all just one red strand. So let's talk about what's going on here. If you notice over here, there is a lagging strand and a leading strand. So remember what we said earlier. DNA replication runs from 5 to 3 prime, meaning that DNA polymerase, yeah, you can rewatch this after the lecture is over. DNA polymerase adds new bases to the 3 prime end. So if this is the 5 prime end of the new strand, the other end would be 3 prime, right? Let's make a note about DNA polymerase. So DNA polymerase 3 DNA polymerase 3 adds new nucleotides to the 3' end of the new strand. DNA polymerase 3 adds new nucleotides to the 3' end of the new strand. So on this leading strand, all is well, right? We're moving in this direction. The old strands are being broken apart by helicase, so it's going to continue in this direction. We can just add one nice solid strand, 5 to 3'. Now, on the other strand, remember, these run anti-parallel. So we have one strand, one old strand that runs 3 to 5. This old strand runs 5 to 3. If DNA polymerase can only add new nucleotides to the 3' end, so the 3' end of the new strand. Let's take a look at this. So this would be 5', 3', 5', that's the 3' end. And that's the 5' end of that strand. If they can only add in the 3' end, it's going to create some problems, right? So it's going to have to create a strand from 5 to 3', and once that 3' strand matches up, like it did over here with the segment of DNA, it has to jump up here and start creating another new strand. So these, this side of DNA replication is called the lagging strand, because it takes more time for DNA polymerase to create these segments or these fragments of DNA, and then it has to jump ahead and create more fragments, all while primase is making more primers. So every time DNA polymerase 3 has to jump, this primer has to be ready for it. So these fragments are called Okazaki fragments. Okazaki is the scientist who found or discovered the fragments, and he described them first, so he gets to name them, and of course scientists like to name things after themselves, right? So these are called Okazaki fragments. Now, when the fragments reach or they reach the old segment here, they aren't exactly attached, and they're not attached because this is, remember, this is RNA, right? Our primase created that RNA primer, and now we have like snippets of RNA backbone, DNA backbone, RNA backbone, DNA backbone. What you see down here is this guy, DNA ligase. Let's talk about him. DNA ligase is a little enzyme, and he glues the Okazaki fragments together. Let's see, ligase glues. Make sure I spell his name right. Okazaki fragments together. So those fragments are going to be glued together by the ligase. So ligase is like our glue. And DNA polymerase 1, which we haven't talked about yet. Let's see, what color haven't I used? Red. DNA polymerase 1 is going to do something before ligase and act on the fragment. DNA polymerase 1 is going to replace the RNA backbone with a DNA backbone. RNA or the RNA primer with DNA nucleotides. So the RNA primer again, RNA backbone with RNA nucleotides. DNA polymerase has to replace that if we want a viable strand of DNA. The DNA polymerase 1 replaces those little segments of primer with DNA nucleotides. And then ligase can come in and glue those meeting places together. They can stitch them together. Okay, questions so far? Let's see, one other thing I haven't talked about yet is the single strand binding protein. You might see it written as SSB protein. You can see it's pointing to these oval purple things. And you might think it's like weird looking or like it doesn't make sense why they're there. The SSB proteins, they help keep the strand apart from each other. So once helicase breaks those hydrogen bonds between the old strand nucleotides, the strand is now open, but it's going to try to form back together. And those SSB proteins are going to keep it apart from each other, keep the strand open, so that DNA polymerase 3 can access those nucleotides. So that's why they're only located here, where there are unbound nucleotides, and you're pretty much going to only find them near helicase. So once DNA polymerase 3 gets close to those SSB proteins, it'll come off and then it'll jump ahead. It'll like leapfrog the next open base, keep it apart. Okay, so let's go back over this really quick before we play quizzes. I'm not going to hit erase yet. But all right, so let's go over it. We talked about helicase and topoisomerase first. So helicase unzips the old strand of DNA by breaking the hydrogen bonds between nucleotides. Topoisomerase prevents supercoiling so that nucleotides are accessible. Once helicase breaks those hydrogen bonds, now we have two strands. One strand is called the leading strand, and the other strand is called the lagging strand. On the leading strand, DNA polymerase 3 can create a new strand of DNA, complementary strand of DNA, by adding bases to the 3' end of this strand, right? 3'. So all is well and good. On the lagging strand, since these strands run anti-parallel, right? New segments of DNA can only be added to that 3' end still, but it's running in the opposite direction. So DNA polymerase 3 has to keep jumping and adding a new strand and jumping and adding a new strand. Now, in order for DNA polymerase 3 to attach to a site on DNA, there has to be an RNA primer. That RNA primer is created by primase. So it's not shown here, but at the origin point of replication, there was a primer here, but it's probably already been replaced by our enzyme that does that. So primase creates that RNA primer. The primer is the attachment site for DNA polymerase III. DNA polymerase III reads the old strand, makes a complementary new strand by adding bases to the 3' end. Once it reaches that end, it can jump ahead. So next, DNA polymerase I is going to replace these RNA fragments with DNA fragments, which creates gaps, and DNA ligase can glue those gaps together to make a nice solid strand. Okay, are there any questions? My phone's ringing. Are there any questions about what we've gone over? Is there any part of like any enzyme or anything on here that you don't quite understand or you need me to explain again? I don't mind explaining something again if you're unsure of what it does. Okay. All right, so we're going to, I'm going to take a picture of this. Feel free to take a screenshot. I'm going to take a screenshot. Okay, so we're going to play quizzes here in a sec. So I'm just warning you that if you're taking a screenshot, say something. I won't exit my screen just yet. Talk about the difference between eukaryote and prokaryote replication. Not a whole lot different. DNA is still going to replicate. There's going to be semi-conservative, so it's going. The new strand will go one old, one new, one old, one new, just like in our other DNA, right? So we have this. This would be our old. And then this would be like our new strand that's forming here. So we'll get two new strands, or we'll get one old, one new, one old, one new. Anyway, so what we're looking at on this slide is a table from, I think it's 14.5, I want to say, chapter 14.5, table out of the textbook. So eukaryotes, remember, we have a nucleus in our cells that contains all of our DNA. Our DNA is also linear, and prokaryotes have circular DNA. Prokaryotes don't have as much DNA that we have, so they don't have, they don't have a lot of DNA to replicate, so it goes pretty quickly. And in eukaryotes, we have much more DNA, and plus they're linear, so our enzymes have to skip around from DNA to DNA, or from chromosome to chromosome to copy them. So in prokaryotes, there is a single origin of replication. And what that means is, remember, prokaryotic DNA is circular. And the way that it replicates is by making like a little bubble here. So it'll start at like one origin. and it'll work its way out away from the origin on either side until we get until we get like a whole nother strand of DNA down there. So that would be our new, that's our new strand that we made. This is our old strand, or, you know, one old, one new, one old, one new. So prokaryotes, they just have a single point of origin, and it goes around the circle until we get a new one, and then it divides. You can also see that the rate of replication in prokaryotes is very fast. So that's one reason why prokaryotes, like bacteria, can replicate and they can divide and make new bacteria so quickly, because they can replicate their DNA so quick. Eukaryotes can't do it as quick, 50 to 100 nucleotides per second. There are different types of enzymes that are used in each replication. So you can see DNA polymerase 1 and polymerase 3 are used in prokaryotes. That's what we've been talking about. But there are other kinds that eukaryotes use. Because there are different enzymes in prokaryotes versus eukaryotes, we can target those enzymes with chemicals, so like drugs, to stop the replication and stop them from growing. So kind of sort of like an antibiotic. Telomerase. Okay, so we haven't talked about this enzyme yet. Telomerase is present in eukaryotes, not present in prokaryotes. For the reason being that prokaryotes, let me erase everything here. Prokaryotes, they have that circular DNA. So they don't have an end to their DNA, it just loops back together. Eukaryotes, remember, we have linear DNA. So the ends of our DNA are exposed. So remember, like during mitosis, the nuclear envelope breaks down, meaning that now our DNA is exposed to other parts of the cell. So a lot of the time it will get damaged by bumping into things or by other chemicals reacting to it. So that's not good, because that can cause mutations and it can cause cancer. But we have some things called telomeres on the ends of our chromosomes that are just repeating segments of DNA. I think it's like 6 nucleotides, just repeating over and over and over. They don't code for anything, but because they don't code for anything, it doesn't matter if they get lopped off or they get damaged. It protects what is inside the DNA, or inside the chromosome here, so the good stuff. Now, telomerase is the enzyme that puts telomeres on the chromosomes, but it's not active in every cell. Telomerase is only active in stem cells. Stem cells, there's another cell. But cancer is typically activates telomerase. So that's why cancer cells can grow rapidly and not get damaged over time. So stem cells, your adult stem cells, and then like embryonic stem cells are where telomerase is active. Adult stem cells can be found like in your bone marrow, and then germ cells. But so every other cell in your body doesn't have an active telomerase. So as these telomeres get lopped off at the end and our chromosomes start to shorten, what ends up happening is that our DNA will get damaged, and that causes aging. So if we find a way to activate telomerase in all of our body cells, we might become immortal. But that might not be a good thing, because cancer could proliferate, and those cancer cells will never die because their telomeres are never getting shortened. So something to think about for the future. So telomerase is kind of that immortal enzyme, but not always a good idea to activate it. Okay, so I think that kind of sums up eukaryote versus prokaryote replication. You don't need to know the differences. Like, we didn't even talk about these enzymes here, so it doesn't. I'm not going to quiz you on those. We're just interested in DNA polymerase 1 and polymerase 3, which we've talked about already. About mistakes during DNA replication. DNA replication does create mistakes, but there are some ways to correct those mistakes. DNA polymerase is one of those major correctors. So let's talk about that one first. So remember, DNA polymerase is the enzyme that reads the old strand and makes a complementary new strand by adding bases to the 3' end of the new strand. It not only does it add new bases to the new strand, but it also corrects errors. So it proofreads what it's done. It proofreads the bases that it's added here, and then it will correct it. So you can see already, let's see, these go together, these go together, these go together, but there's already some errors here, right? So it's going to correct those errors, and then hopefully the new strand will be okay. But if the new strand still has mistakes in it, there are some other ways that those mistakes will be corrected still. So DNA polymerase is kind of the first line of defense for correcting mistakes. Oops, defense, if I can spell. Okay, so DNA polymerase, this is polymerase 3, is the first one. DNA polymerase 2, DNA polymerase 2 is another one that will help correct mistakes, but that happens down the line. Hopefully we don't get to the point where DNA polymerase 2 needs to be activated. DNA polymerase 1 will also help correct mistakes. Oh, I only put 1 and 2 up here. DNA polymerase 1, 2, and 3. DNA polymerase 1, remember, 1 helps with removing RNA primers with DNA. So as DNA polymerase 1 is removing those RNA primers, it's going to double check that the base pairing is correct. Okay, so another mechanism that can happen is mismatch repair. Mismatch repair happens after the strands have been created. And you can see here that mismatch repair will correct a base pairing. So a lot of the time when we get mistakes, what happens is we get like a bulge or like a divot in our DNA. So remember, we have four bases overall, which are purines, and then there's pyrimidines. Purines are the big ones, and those are adenine and guanine. And then pyrimidines are thymine and cytosine. So first, if there are like two purines that match up, like if A and G were here, let's just make this an A for right now. If that were an A, there would be a giant bulge in the DNA strand, and that's going to cause mismatch repair to come in and correct it. But you can see here that there's an A and T, or an A and T, a T and a G. So that T and G, they don't normally match up. They're going to kind of be distorted because the hydrogen bonding isn't working correctly. And so that'll create a bulge in DNA as well. And then mismatch repair will come in. So when we have a mismatch, like in this case, A and G or G and T, it creates a bulge or it creates a divot in DNA. And then mismatch repair will see that, or quotation marks, see that bulge or divot, and it'll correct whatever's gone wrong. So hopefully the mismatch repair will correctly place the correct base in there. So it replaced that T with the C. And if you look on this strand, let's see, C, C, G, it should have been a C, right? Right here. So that's correct. And then you can also see it up here too. But sometimes mismatch repair does not put the right base in. So let's say instead of replacing the T with the C, let's say it replaced the let's see, we had T, so let's see if say it replaced it with an A. So that's going to create probably a new protein. Or maybe it doesn't create a new protein, or it just creates a new amino acid, which could cause mutations, could cause problems. So hopefully mismatch repair does it correctly, where it replaces the T with a C in this case, but sometimes that doesn't happen, and it's the opposite base that gets replaced, which causes mutation. There's another thing called nucleotide excision, and nucleotide excision helps to repair what's called a thymine dimer. So thymine sometimes reacts and bonds with itself in DNA, and that creates, you can see, like a weird bulge in DNA strand, and that could stop DNA polymerase 3 from copying the strand correctly. So DNA nucleotide excision removes that weird bonding so that it lays flat again. So there's a, if you want to look in the textbook, there's a gross picture in the textbook of showing you the skin lesions that could happen when there's a lot of thymine dimers in your DNA. And thymine dimers can happen when you're usually like exposed to UV light. So if you have sunburn over and over and over again, your DNA reacts to that UV light, and you get these dimers, like mutation types, and that creates like these lesions on your skin. Pictures in the textbook if you want to look at it, but I saved you that. I'm not putting it in the PowerPoint, but it's kind of gross. So UV light is the biggest cause of those dimers. Okay, so let's see. We already talked about telomerase and telomeres, but just to show you visually what a telomere is, it is over here. So that's telomerase. So here's a telomere. So GGTTAG. And you can see GGTTAG. So it's just a 6 base pair, just repeating over and over and over. It doesn't code for anything. So if it gets lopped off, no big deal. But eventually, enough of it gets lopped off that it starts damaging DNA, which causes aging. Okay, so usually when a cell runs out of telomeres, it starts to die, which causes aging as well. So back in 2009 is when we actually figured out what telomerase does. So not too long ago there, and we're still making these crazy scientific discoveries. Okay, so we've already gotten the mutations. Cool. So let me show you. I'm going to do a poll question really quick before we move on. Okay, there's that. So the question is, a scientist randomly mutates the DNA of a bacterium. So we did it on purpose. She then sequences the bacterium's daughter cells and finds that the daughters have errors in their replicated DNA. So the parent bacterium likely experienced a mutation in what enzyme? Okay, so most of you said B or D. So B or D would be correct. B, well, I would say D would be more so the correct answer. DNA polymerase 1 is the enzyme that's replacing those RNA primers right after DNA polymerase 3 comes through. So I guess if we're talking about like the sequence of DNA fact checking, it would be DNA polymerase 3 followed by DNA polymerase 1 and then DNA polymerase 2. So DNA polymerase 1 would have caused a lot of those errors if we were just picking from those choices. All right, cool. Let's see, let's get back to PowerPoint. Okay. So mutations, we already talked a little bit about mutations in DNA. So a lot of the mutations are point mutations, meaning that there's just one base that's incorrectly copied through DNA replication. Sometimes there's more than one base that's inserted or deleted, which causes a frame shift. So let's talk about point mutations first. Point mutations can be silent, they can be missense, they can be nonsense. Silent point mutations, like a substitution. So let me see, I'm going to type substitution where it was in A, and we accidentally put a T in there. A substitution really doesn't cause, or this kind of substitution didn't cause any damage. So it's still coded for the same protein. A missense point mutation would cause a change. A nonsense mutation would cause the protein to get cut off. So point mutations, or like a substitution in this case, are the most common errors in DNA replication. And substitutions have caused most of the changes in population over time. Let's see, I'm trying to think of one. So cystic fibrosis is caused by a substitution. So just one base pair is off, which causes the disorder cystic fibrosis. So substitutions are very common. I think I want to say insulin is if you have type 1 diabetes, I want to say the insulin gene is also substituted, or a wrong base pair is substituted, which causes a defect in the insulin gene. I want to say that's correct, but don't quote me on that. But definitely cystic fibrosis. So most common in the population causes the most changes over time. There are some things like frame shift, which causes a change in every protein or every amino acid down the line from it. So those are usually caused by like an insertion or a deletion. Insertion or a deletion. So we deleted T and A from this first one. So those every base pair after that moved over 2 spots, which causes a frame shift mutation. And a reading frame is typically 3 nucleotides long. So if we delete 2 of them, that's going to change our reading frame, right? So typically frame shift mutations are really, really bad, and that causes a huge change in the resulting protein, which might cause like death of that cell or death of that, I guess, fetus in case of replication or case of reproduction. So frame shift is the most deleterious, but if we have a frame shift of 3 nucleotides, it would not be so bad. And I don't think I have a picture of that. But I guess if we deleted G, T, and A, it really wouldn't cause too much of an issue because we're only missing 1 amino acid. Probably won't cause too much of a change. But yeah, I think you get the point. Okay, let's go on. These are all the different types of mutations that we could have. We're going to look further at these next week when we talk about how we get protein from DNA sequences. But first, we're going to look at some DNA sequences, and I want you to tell me what types of mutations we have. So I'm going to write them at the top here. We could have substitution, we could have insertion, we could have deletion, substitution, insertion, deletion. Yep, those are the only ones. I don't think I put anything else in there. No, okay. So if we're looking at our normal DNA, this is the sequence that it has. CAT, CAT, CCT, AAG, GTA. So let's say our DNA gets mutated, and after the mutation, it looks like this. So what type of mutation would this be? Or what, yeah, so it would be substitution. So and it happened right here at G. So instead of CCT, it was CGT, which could cause a new amino acid in that case. But that doesn't change the other amino acids, right? Okay, so let's look at the next one. Mutation 2. Where is the mutation and what is it? Yes, so we had an insertion. Insertion, good job. And mainly you could point it out because there's an extra one here at the end. So where was that insertion? Right here again, right? So we put a G in instead of the substitution like we had earlier. So that causes a frame shift, right? So now every amino acid after that point, we had AAG, now we have TAA, which probably codes for another amino acid. And the same thing, we had GTA, now we have GT. Okay, so last one is what? Yeah, deletion, so we're missing one here, so that's a big giveaway. Deletion, where was it? right here. There should have been an A. Right, yeah, so there should have been an A, and now if you look, there's a frame shift. And now all the the frames have changed. The amino acids probably changed. Okay, cool. I think that kind of sums it up. There is a really nice TED Talk that goes over like Neanderthal DNA and how we can look at Neanderthal genome and compare it to ours, and we can see like what mutations have happened over time since the cavemen ruled the earth and now we're here. So there's a really cool video that goes over that if you want to look something about genetics. But you don't have to. Totally up to you. All right, so that is all I have for you today.